Aurelia, Fatma was saying hi.
Oh, hi. What's up? Hello, Ben W. What's up?
She's here now.
Oh, hi, Fatma. Um, so I tried to launch the Hangouts on air, and I think it's working. So if anybody wants, if some for low bandwidth reasons you can't join for video, check out online 25. Hopefully the YouTube link is displaying the Etherpad. That's really what we're hoping for.
Hurry.
Uh-huh.
What room is this? This is Caitlin's?
Yes.
Okay.
I hope this is the right one that everyone got here, I think, so it's okay. I must have publicized the correct room.
Yeah, that's what the link that was in the calendar.
Okay, good.
Yeah.
All right, good. Because I was like, oh no.
Sam and I were both in your room going, wait, where's
Oh
everybody?
dear, I'm sorry.
No, not your fault.
So for people who don't know, video has multiple rooms for every Mozilla employee, so you can tap into anybody's personal room and chat with them, and sometimes like photobomb meetings that you didn't realize were going on. So we're currently in Caitlin's room, who's director of the Mozilla Science Lab. She is on break, so she will not be joining us. But yeah, also for those who just joined, there's a Hangouts on Air link. We're recording this session. If you don't feel comfortable being recorded, your face isn't recorded, so don't worry. It's just the Etherpad, and whatever you say, hopefully the audio will record. We won't really know until the end, and sometimes it doesn't record. So apologies if you intend to tune in later and all you get is like a scrolling Etherpad. But yeah, Rob, I think we can kick it off whenever you're ready.
Yeah, perfect. I think we're ready to go. Thank you, Rita, for asking me to MC. My name is Rob Schaefer. I'm at the University of Minnesota. But we can just get right into it. So today, really said we're broadcasting this on Hangouts on Air, so that'll be available later. Looks like we got about 9 or 10 people. If you haven't signed in on the Etherpad, just put your name and affiliation, and then Twitter, GitHub, different handles. Online 55 on the Etherpad. And also, if you something interesting in the past month, there's a spot to share that on line 60. Matt, the agenda today, we're going to talk about Julia, specifically BioJulia. Julia is a newer programming language. It's about 3 years old, and it's geared towards open source, open science, and then also performance. So we have some great speakers to talk about that today. So that'll introduce Ben and Richard. They're going to talk about BioJulia. So you guys take it away.
Yeah, so BioJulia is a project that Ben and I and a couple of other people started. Was it 2 years ago now, Ben?
Yeah, it would have been about beginning of my second year. So yeah,
2 years. Your PhD. Yeah. So the idea of the project is to build up the tooling to do bioinformatics in Julia. And at the time, there really wasn't anything coherent available for Julia to do bioinformatics in. We don't particularly see our leaders in the field or anything, but it just seemed like the thing didn't exist. We wanted to use it, so we started making it. And we got pretty lucky, I think, early on that, Ben, who was already doing some really cool evolutionary analysis using Julia, Daniel Jones, who is just a magician in the Julia language. Those two were willing to help me start this project. And in fact, I say help me, they've written almost all the code and I've done nothing. Sit back and help help the conversation keep going in the chat room and that sort of thing. Ben, do you want to talk about like the higher level goals of the project, like what we care about?
Well, when I start, for me, the goal is basically to improve science. I think that's the big goal that we cared about at the start, because I only really started doing bioinformatics and programming as I transitioned into my second year of my PhD, and I soon discovered that methods in a lab are treated really rigorously and are shared properly and are published properly, data is published sort of properly, although depending on properly, I mean there's paywalls and all this nonsense. The code was really sort of written in a hurry because there's a deadline, and then they shove it on their GitHub. It's probably got errors and only worked for them. And if you wanted anything sort of high, you'd write in your scripting language. And then if you wanted to write anything more powerful, you'd have to link in C or C++. And what I discovered was I spent most of my time, I'd gone from spending most of my time thinking about biology, and now suddenly I'm spending all my time thinking about computers. Which is not good when you're trying to get a PhD in biology, right? Or do any biological work. So for me, and also when you wanted to understand how a bit of software works, because it is a method, that's all else. It's much like some dilution method in a lab or some DNA extraction method in a lab. When you're actually trying to understand it, and then you have to write all this C and C++ code, or you're publishing a method and a reviewer has got to read and write your C++ code and your R code and your Perl code, and they've also got to be an expert in the exact same field as you. That's really not good for understanding science and is really bad for reproducibility. So along came Julia, where you could get the performance that we want, but you can express everything in a higher level language, and it's immediately understandable, almost immediately understandable to anybody who's ever used Python or MATLAB before. So we thought, wow, we want bioinformatics in this. Like we want bioinformatic libraries in this. So really the goal for me for this project was I never want to have to look at any lower level languages ever again, because I want to think about biology and improve code. And you know, when biologists look at some code that's high level, or you tell a biologist you have to program in this high level language, and it's also really fast, they're more likely to learn that. And if you go, oh, by the way, you have to spend about 2 years learning R. Oh, and now now that's not really good enough. So now you have to learn C++ as well, and how to bridge those. Oh, and by the way, some people still use Perl for some reason, so you better learn that as well. And if you're an environmental modeler, some people use Fortran as well, so now you have to learn this. And you're like, no, I don't want to learn all this. I'm a biologist. I want to know what's happening in the organism, not what's in my machine. So that was really the goal for me starting out BioJulia.
So the classic problem that Ben described, we call the two-language problem, although it can be more than two. But that's where you have some idea and you want to see if it works. So you write it in something that's easy to write in, like Python or Ruby or JavaScript or whatever. And once you figured out that it works, now you need to make it perform well on bioinformatics data, which is huge. You know, we talk about gigabytes of data in the smallest analyses that we do, terabytes or more in large experiments. And if your language isn't fast enough or your code isn't written to be performant enough, you have to rewrite it. Otherwise, you're never going to get the results in the lifetime of a human being. So that problem, where you have to basically be an expert computer scientist as well as being an expert biologist, is, I think, something that we both felt during our PhDs. And it turns out, talking to lots of other people doing things that involve analysing biological data, that it's a widely felt pain. So that's what I agree that that's one of the things that we wanted to solve. Julia has the potential to be that one language, easy to read and write, and it performs at an incredibly high level that competes with things that are really quite difficult to read and write. And then I would say that also at the beginning, we talked a lot about what we wanted the community to be like and how we wanted the project as a whole to work. And it was really nice to be in a team where everybody agreed that we cared a lot about inclusiveness, that we cared a lot about bringing everybody with us, and we also wanted to not rush anything. We wanted to put out something. We haven't made a fuss about our releases yet. We're working slowly and very carefully towards having something that should really work well for people, I think. And we've got you go ahead.
There's this I've seen this thing where almost every language has a bio project. So you've got like BioPython or BioPerl, BioConductor, and when you read some of these, you kind of can see, especially the newer ones that pop, like because a lot of languages don't actually stick around. They have to reach a threshold to stick around, right? And people, when a scientist sees a new language, or a bioinformatician sees a new language, I think that they very, very quickly come out with some library for doing bioinformatics or a bioinformatics thing in that language, so they can go, great, I'm here first. And basically, it's almost like a carbon copy of whatever was in Python or whatever was in Perl, because they just want to write it out in that language and then just get it out there and maybe publish a software note. So from the start, we really wanted to make sure that we thought about the things we're writing very carefully and write what's best for Julia and just ignore what everybody else is doing. And that has led to the sequence module being rewritten twice now, I think. The phylogenetics module, which I'm finally starting to merge, has been rewritten at least 3 times already in order to take advantage of this. So in the phylogenetics module, for example, I was learning Julia, and I used R a lot. So I took a lot of ideas from how the R packages worked for doing evolutionary analysis. And then we passed around some more, some people looked at the code and came up with, hey, well, Julia lets you pass composite types by reference, which never happens in R without some serious black magic. So you can just have phylogenies as like these linked nodes. I was like, oh, that's fantastic. That's really good. We read it all. And then finally did another review to merge that. And then Kenta, who's also a really talented guy who we took on a few months after we sort of started. I think it was as a Google Summer Code. Now he does lots of things. And said, well, now they've made light graphs, a package called light graphs in Julia, which is basically those people who are far better at graph theory than I am, come up with these really efficient types of graphs and things like, okay, so rewrite the phylogenetics module again to just take advantage of those graphs then, so I don't have to rewrite all the linking structures and the path traversals and the iterators and the searches through a tree all over again from scratch. So it's taken, as Rich said, 2 years to get to the point now where we're getting a critical mass where more biologists are starting to get interested and are lurking on the chat room and looking at what's being developed. And our scientists say, okay, now I can kind of use this for stuff that's actually useful in my day-to-day work. So we're having to care about, in the last few months, I've had to care a bit more about the website and the documentation and this kind of thing, because people are going to start reading it. And we've had people submit modules. So one guy submitted a wrapper for a program called BLAST, which is a very common tool for biology. Also, somebody else submitted a module for working with protein structures, which I'm an evolutionary biologist and sort of geneticist, so I don't look at protein structure at all. So that was really useful. So yeah, we're quickly approaching a point where more biologists are going to jump in and start adding more and more and more to the project. Whereas for the past 2 years, most people have been going, it's a big risk. Is it worth it? What if the language doesn't stick around? Now it's clear that the language is sticking around. The functionality is starting to become complete. We're getting more interest.
I didn't want to publicize the project too much initially because we weren't ready, and we knew we weren't ready. We wanted to do that sort of slowly building the right thing. And Ben mentioned that we've deliberately not ignored, but we've looked at what people have done in other languages. We've thought about whether that's the best way to do it, and then we've redesigned from the ground up if necessary. But the other thing that we've done is that it turns out that every time a new language pops up and gets its own bioinformatics library, people do a ton of things from scratch, or they develop a bunch of resources that really we should all be sharing between all the languages. Things like bioinformatics has dozens, perhaps hundreds, or even thousands of file formats that have been designed sometimes quite carelessly, but are heavily depended on by biologists in their work. And every language that we looked at that came before us, the people making the bioinformatics libraries had just written parsers from scratch. They'd taken a bunch of random examples of files from the internet and used their test cases. And it was all a mess, and it leads to a situation where if you try and do something with a file format in one language, you get different results than if you try and do the same thing with it in another language. So we've been trying to fix these sorts of systemic problems as we go by creating universal resources for doing this kind of stuff. So, for example, we have a GitHub repo where we collect specimens of bioinformatics formats, and then we have a bunch of metadata about those files so that anyone in any language can use those files as their test cases. And if we're trying to get to a stage where all the bioinformatics projects have a single library of test specimens for these formats that are examples of how they appear in the wild and all the weird and wonderful things that people put in them in the wild, so that we can all make sure that we have consistent behavior across the languages. Then we went even further than that, and this is was a project of Daniel Jones, who, as I said, is a wizard, and he brought into the project this idea of using Ragle, which is a parser generator format, roughly. It's a way of describing a parser in such a way that any programming language can generate an efficient parser in that language. And so what we've been doing is building a library of these language agnostic parsers in Ragle. I don't know how it's pronounced, I've only seen it written. But building up this library of parsers that can be used in any language that has a Ragle compiler. And again, this is trying to just get to a stage where we can all collaborate on the resource across all languages, because there's a lot of people spread out across a lot of languages. We can get to a stage where we're all collaborating. I think we can be much more efficient, and to a stage where everything behaves consistently across languages. So yeah, we've got a lot of. I would say that as a result of all the work we've put into making things very Julian, like very optimized, to be the best that they can be in Julia. And as a result of some of this work we've been doing to make things shared or as shareable as possible across languages, the project has probably seemed a bit impenetrable to people coming at it from the outside, because we have a lot of complicated technology in there. So a big challenge for us now is how do we document that stuff, make it really, really easy for people to join the project and contribute code, and make it really just trivially easy for people to use it. And so over the next few months this year, really, we're going to focus on getting that stuff and getting a website which helps people just by using BioJulia without breaking a sweat, we hope.
So I have a question about that, Richard. Do you think that there's a trade-off there between trying to make things generalizable or reusable versus a lot of times most experiments in science are really direct and they have special cases. And so is that scalable? Sort of dropping in modules like that?
Yeah, I think we have to really pick our battles. So there are some things which are common to every experiment or most analyses. And those are the things where we want to try and make sure that we're all sharing the effort. But the thing that, the more customized something gets, the more specific to a particular kind of analysis, the less worthwhile it seems to try and put the effort into making things generalizable, I think.
So have you focused more on your guys' own PhD work and then trying to make that generalizable versus trying to make tools that everybody can use, including yourselves?
We've got a bit of a balance of those two things. So we did an initial design stage at the beginning of the project where we spec'd out what for most modern bioinformatics people would need to have in BioJulia. And so we have all that stuff spec'd out, and we're working through completing all the things in that list. But the things that have taken priority or that have appeared first are probably the things that we've needed in our own analysis. So for example, Ben's work on phylogenetics would probably not be the first thing that most people would implement in a bioinformatics library. But Ben's doing bioinformatics, and it turns out so are dozens of other people who have come along and looked at BioJulia, and that's been a hook because people are doing bioinformatics. There's already an expert on the team. They can see how it's being done, and they're doing phylogenetics. There's already an expert on the team. They can see how it's being done and talk to Ben live about it. Not being productive straight away. So yeah, there's a balance, I think. To have an input.
So the structure of the project, I think it's a bit of a balance because we have the organization on GitHub, and then we have one flagship package called bio.jl, which has most of the core stuff that most people want to use in a couple of different fields. So whatever software I'm writing, whatever special case I'm dealing with, or whatever project I'm working on next in biology, I will almost always certainly probably want biological types of biological sequences. I at least, and some other researchers, but not necessarily all of them, will want phylogenetics. They'll want types that model certain population genetic scenarios and the equations that go with them, and so on. Some scientists, a certain subset of scientists, will always want tools for dealing with protein structure. In fact, I might even, if I'm looking at the evolution of proteins. So, and these are all in their own submodule under the bio module. So these are the core things that most life scientists would want, either in their work or if they were going to build some other specialized application. Those cover all the basics, so they don't have to rewrite anything from scratch. So if you're writing some sort of new clever algorithm for dealing with, for computing some magic stat on sequences, well, you don't have to rewrite all the code for dealing with sequences because sequences are in bio. And then your really, really obscure project-specific algorithm you came up with, well, that could be another package that uses the bio module, imports it, and then you could submit that really specific package to BioJulia, and it will be a separate package. But it's the core dependencies as well as the package itself will then be under BioJulia's stewardship then, and that guarantees that your code will be maintained and have a long life, and we'll always have somebody to look at it and make sure it's okay. Which is another thing. People, after a couple of years of creating their algorithm, publishing the paper, and then sticking it on GitHub, don't care anymore after that, right? They might maintain it a bit if it's a really important tool for their research, and it's a long standing thread of their research. But if it was just one project, then it just gets left and forgotten about. They don't maintain it. Scientists don't want to maintain things. So we've come up with a structure for how code should be submitted to the organisation that we've got in our contribution guidelines that help us strike that balance between making sure that everybody's singing from the same song sheet on the really, really important stuff that everybody needs, and then at the same time allowing you to branch off a little bit and do your individual specialised work.
You reminded me, Ben, that there's another way in which we try to do things that are kind of generalized. So as Ben said, we've got the BioJulia organization on GitHub, and bio.jl is the main package. And we have a ton of other packages, some of which are biological, but actually we've written a load of packages that are just improvements for Julia in general. So a bunch of data structures and algorithms that are used in all kinds of other Julia projects now. That we release as separate packages, and this is something we try to do as much as possible. If we find that we are inventing something that likely has a use outside of bioinformatics, then we make that a general package and release it back to the Julia community. And actually, I think a lot of people have heard of the project that way. We get a sort of steady stream of Julia programmers coming into the chat room and joining in, who have found out about us because of our compression libraries and
I think libc's been the big one. Which the bio package imports, and there's some other things like indexable bit vectors, which are incredibly important for bioinformatics, but at the same time, loads of other people want them. It's not just for biologists. So bio covers. So we have several packages that are generally useful in computing. But just also happen to be really important for biologists. They're all imported into one focal point in bio, which then all adds the truly bio-specific stuff, sequences, phylogenetic trees, proteins, and so on. And then from that one baseline, scientists can then branch out and write their own individual packages and their own methods. That's kind of how
questions. There are a couple of questions in the Etherpad.
Yeah, we can move on to questions. I think you answered the first one. I'm Brown, I think, on the Etherpad. Second one is I'm totally new to Julia. What should I know about its special quirks and superpowers? So can you, having some experience and implementing modules yourself, can you give me advice there?
So the really special thing about Julia is the fact that it has a dynamic type system, but it also has type inference that's coupled with multiple dispatch. And so what that allows you to do is write really high level code. And then compiler will basically figure out what the concrete types will be when it gets compiled. And it's a JIT compiler. So this allows you to create functions and overload functions with lots of different argument types, and then different behavior will apply to those different sets of types. So, for example, you will just write print. Say you want to print some number. Say you want to print a string, and you type that in the Julia console. What will happen is there are actually lots of different prints. There are loads of different print methods, and what the Julia compiler will do will figure out what type's being input into is the input of the function. They'll go, right, okay, this is a string, therefore we need to use the version of print that is specialized for the string type. And the Julia compiler takes the specialized function, the print function for string, and then compiles it. And then then you could just type in the console print, and it'll be a DNA sequence type. and the Julia compiler will do the same thing. It will infer. It will go, okay, this is a DNA sequence. We need the version of print for a DNA sequence, and take that specialized code, and then compile that and run that. So basically, it's kind of like you have all these different versions of functions, and Julia looks at what types are being put into the functions, and then it will see also what types are coming out of the functions, what gets returned, and it generates then really efficient code, because it's sort of, even though you're programming in a very dynamic way and it's on the REPL and it's all interactive, it can pick the most efficient code, compile it, and it gets compiled once. And then if you say you use print on a string again, it goes, all right, we've used that before. This is the compiled thing for print string, and it's because it's been compiled, it's fast. So this is really cool if you're looping over arrays and things.
It's a bit like,
you're doing big array. If you're doing big array operations. Because it'll get the function gets compiled once, and then it's being looped, and it's just being applied to say every element in an array, and that's really fast thing, because it's just reusing the cached compiled version.
It's a bit like writing Python and getting C code run for you automatically, something like that, just in case people are not following what types are and things like that.
Yeah. What I also really like about the dispatch thing, the multiple dispatch system that Julia has, is it's used by Python or similar language. You don't have a lot of choice. It's object oriented, and object oriented is a very good way of writing some code. It's a terrible way of writing all code. I can't remember who said that. I'm paraphrasing somebody. And in science, this happens to be true, because sometimes types and analysis types that you deal with do actually fit into like a hierarchy, but sometimes they really don't. And if you want to extend some code that another scientist wrote in some other, as part of some other paper, and you want to build on their methods, trying to cram something into some hierarchy can actually be like quite a big pain. With Julia, because it's all based around types and then methods, and the two things are sort of decoupled. Your types are your data, and your methods are the things you do to the data. Because programs are built around that, if you want to extend somebody's methods, just create your own package that uses their package, so it imports it. That brings in all the types and methods that they've already written. Thank you very much, scientists from 2 years ago. And then you just write your new algorithm that just uses all those types, that uses those types and just computes things in a slightly different way based on your improvement on their research. So I think
you can replace any function or type that they've already defined with your own more specialized or efficient version.
Yeah,
but it'll automatically use all the previous stuff in
exactly.
a way.
Yeah, so
I think related to. Go on.
No, you can.
related to that point, because the question was, what should we know about the special quirks? So because of all that cool stuff that Julia can do, you have to be a little bit careful about how you write your code to enable Julia to be able to figure out the most effective way to generate the compiled code. So if you write your code in such a way that it's ambiguous what kind of type a thing might be, in a function, so you have a variable and it could be either a string or a number of some kind. If there's absolutely no way for Julia to guess what it's going to be, then it's going to be much less efficient at runtime than it would be if it was just a bit clearer. So that means that it, in Julia in general, and this is true of all programming languages for good philosophical reasons, but in Julia, for performance reasons, you should break down your functions to small units that do one thing very efficiently or very explicitly, and that really helps the compiler generate code afterwards.
Yeah. So don't use functions don't create functions that might, depending on the value. Let's say there's a function that accepts a floating point value. If the value is less than 5, it returns a string that says, yay, I'm less than 5. But then if it's above 5, it returns a Boolean value. That would be a bad idea, because the function could return two completely different things, which doesn't lend itself to sort of static type analysis. It's not predictable. The compiler doesn't know, because depending on what happens at runtime, you're getting different types, and that's not optimal. But the great thing about that is, though, even if you did write a function like that, it would actually still work. You can still run that. Sure, it just won't perform as well, because Julia is still a dynamic language when all's said and done. So the really nice thing about this is you can prototype stuff and just get stuff working, and then it's okay, great, I can write a paper on this method works, and then later on you can optimize and make it faster. Whereas before, with another language, that would mean prototyping it, getting it to work, and then going, great, now I need to optimize. Oh, better crack out the C. Whereas optimizing in Julia just means reading your Julia again and just being a bit more careful with your functions and your code, and making sure all your types are predictable and so on.
So since Julia's a younger language, how are the developer tools like debuggers and profilers to sort of get at this efficiencies? How mature are those in your experience?
So I can't remember what the state is of a debugger. I think it's being worked on. I don't know if there's a version that already works and exists, but there is a profiler in. Is there?
Yeah.
And
several.
There are a couple of profilers. There's a sampling profiler, isn't there? Where you run your code and it takes sort of the location where it is in the code at various time intervals, and then based on that you can see where the sort of bottlenecks are in your code, or where your code is spent, which function is taking a long time. The Julia, you can actually make the Julia REPL return to you what the at various stages of interpretation and compilation, what code is being generated, so you can get the abstract syntax tree out of a function. You can get the just in time what's it called? Clang? No. The instructions that pass to the just-in-time compiler to generate LLVM. That's it. That's what I'm thinking of. The LLVM instructions, or you can go all the way down and make it return the assembly instructions. So you can, if you can read assembly and you understand it, you can look at a very low level and see exactly what's happening. What you're generating.
All of those things that generally come under the heading of metaprogramming, in that in addition to making the computer do something, you can access how it's doing that thing in the internals of the language at the same time as it's doing it, which sounds ridiculously complicated and perhaps a bit like something you wouldn't want to do, but it actually adds all kinds of powerful stuff. It means that people can build those developer tools really easily, and that's been designed into the language from the very first release. I think that they've been trying to make it easy for people to do stuff, debuggers and compilers. And as a result of that, there are two at least really nice IDEs. So it's like a piece of software that allows you to write Julia, and that's focused on helping you write Julia. There's one called Juno Lab, and there's one called Julia Studio, which is named after RStudio, but works differently. And both of those.
Could you say those one more time? Sorry, I missed them.
Yeah. There's Juno Lab, JUNO LAB, or Juno, is the normal name for it, and then Julia Studio is the other one. I'll write them in the other idea.
And is there a kernel for Jupyter Notebook?
Jupyter Notebook. The Ju stands for Julia. So
yeah, I just thought it's probably mentioned.
Yeah, that's it. So it used to be the IPython notebook, and people would make custom kernels, and there still are loads of custom kernels for Jupyter, which is the descendant of the IPython notebook. But now the three main scientific computing languages come with the project. So Jupyter, Python, and R are all catered for by the core of Jupyter notebooks.
Yes, C-Line, C-Line 74. There's a podcast about this.
So that kind of sort of leads us into a question from Alex. If you're on the call, so if you want to ask, you can.
Yeah. So I was just wondering, because obviously you write, and the scientists they'll write their code for the particular analysis, and then they'll publish it, and then they'll leave it on GitHub. Is there a good way of getting them to actually, encouraging them to actually submit this as like a library or a module, so that someone else can then use that properly, as opposed to having to figure out exactly what they've done in their publication or whatever?
There are at least two things that we will do. The first is that we need to make the tools as easy as possible. So if it takes a lot of someone's time, or it takes a long while to learn how to do that, people will not do it. Scientists aren't paid to, well, they're not paid very much in the first place for anything, but the little that they are paid is not given out based on how much someone's done reproducible analysis. It's based on other things, unfortunately. It's a separate thing that we can run to our friends. But to take account of that, we have to make it really as technically easy as possible for people to behave well, to make reproducible analysis, for example. And so the tooling that we need to make really good, and then we need to make the documentation and things like the community welcoming and easy for people to get involved in.
So one of the things we want to aim for is make it as easy as possible. So we have reasonably clear instructions already on the website about how to contribute, either to existing modules and packages, or if you want to submit an entirely new thing with explanation of what the expectations are and what the guidelines are. But I remember writing this explicitly in the documentation. If it's gone, we should put it back up there in that documentation, that if any of it seems daunting, or they're like, oh God, that's a lot of guidelines. Do I have to? If they just come to the community on one of the chat rooms or through an issue or anything, like as long as they reach out to us somewhere, ideally on the chat room, like one of us as the sort of core developers and maintainers, or somebody else in the community who's just nice, because I think we have got lots of nice people. We'll just really help them with it and go, okay, you want to submit this, great, let's, and we'll actually help them with it, because not everybody likes to just be told to RTFM. And even though they read the documentation, people are nervous. Like they go, I understand what the guidelines are, but how people doubt themselves a lot in coding. It's kind of an experience thing. You want somebody to sit there with you the first time you do it and take you through it step by step and sort of be friendly. And so that's what we intend to do. So all the packages and submissions, pull requests go through 3 stages. There's a planning stage, a work in progress stage, and a review stage. And the planning stage can begin when somebody goes, I'm going to be working on this. It's a fairly cool idea. And then people can just jump in with comments and say, well, the best way I think to do this in Julia would be whatever. And then the work in progress stage is where either they're just working on it on their own, or maybe in conjunction with somebody else, because the result of the planning stage, somebody else might go, yes, I want it, I'm going to help you out. Let's do it. And then they'll be pushing and pulling to the same repo. And then comes a review stage where they'd like to merge it and they want to get comments from as many people as they can to look at the code and go, well, you could have done this a bit better. But in the review stage, crucially, we say in the documentation, and we want to encourage this in our community, is rather than just say, go away and fix this and come back, if it's something that is easy to do and is 5 minutes, pull the branch, implement the fix, and help somebody out, especially if it's their first time and they don't really know how to use GitHub or Git or anything. Like, we want to help walk them through it as much as possible. Like, can't speak for everybody, but for me, that's what being a maintainer is kind of about. It's no good to just tell people it's not good enough. I mean, because you want the functionality in your packages at the end of the day. And if you tell people no enough times, they'll just go, all right, well, I'll stick it in my own repo, or I'll go and find somebody who will have it.
A core tenet of the community is that we expect people to be nice.
Right.
Not just like,
and understanding, because most bioinformaticians either start out in computer science, in which case they're all down with this anyway, and then they learn biology as a kind of secondary thing, and as a result they're really limited in the biology they understand, which is a flaw. Or the biologists who come to computing and programming the other way, and so they write code that's really not efficient, they don't understand core programming principles, they're not comfortable with Git, they're not comfortable with the command line, and they just want to do the bare minimum and understand the bare minimum in order to do the biology that they actually understand really well. So more than just being nice, being understanding of the position that biologists are in, because we've all been through that shock of being thrown in at the deep end of programming something, and then like thrown in the deep end of having to upload your stuff to Git because it's some requirement to the journal that it has to either be on GitHub or Data Dryad or somewhere like that. And just remembering that you were a noob once as well is really, really important.
Definitely. I think there's a couple more questions, but just in for the sake of time, if you guys could go check those out on the Etherpad and maybe fill in where you can. Should probably move into the rest of the call. Study group updates. Aurelia pointed out it's pretty lacking right now. So if people want to go throw something in there quick, I guess I could start with a verbal update. The end of the semester sort of came and people were busy with other things, so it's been a couple of weeks, but I sent out an email today. We're going to meet again next week here at Minnesota, and we're going to decide on what we're going to do for the code sprint coming up. So hopefully going to form a team and decide what projects to focus on and sort of switch gears there a little bit, and then figure out what we're going to do for the rest of the summer. If anyone else on the call wants to give a verbal update on study groups, feel free to chime in. Anyone? No?
Maybe
Sort of the
Sarah?
same
Sarah,
situation.
I don't know if
elsewhere?
Sarah's verbal on the call, but she just put a thing in the side that was like, we're disbanding for the summer, so not much to update at UW Madison. So I'll put that in the Madison.
Are there other people planning on doing the code sprints? Yeah, good.
yeah,
A lot of fun last
I'm
year.
going to the one in London.
Perfect.
I have a question for anyone who's on the call but hasn't put an update in, or maybe we could put this in the Etherpad. If you're not disbanding for the summer, do people have suggestions or ideas for things to keep things for like little plans or strategies to keep things going over the summer a bit? Like, would love to know what's been successful in the past or what you're planning to try, or if we could help you guys in any way, support you guys in that.
Yeah, for sure. I think we sort of decided to try some new stuff. So we'll definitely let you know how that goes here in Minnesota. You can check out some nonverbal updates on line 128 from Madeline and Akita. Seems like they are deep into creating lessons. Looks like we have some lessons on Hangouts on Air now. So that's really cool. And then also see lessons here for Docker. There's one for Julia, R. So some lessons that are going up at Bristol and CERN. Other new lessons, pandas for data analysis from Derek. Some more Git stuff on branching from Luke, from U of T. What else do we have here? Some tutorials and topics. These look like some more commentary on Julia, and some tools and specific YouTube videos.
Oh
As
yeah,
for
Rob, do you mind if I
announcements,
chime
maybe
in about
Aurelia,
those?
you wanna? Sorry.
Do you mind if I chime in about those resources?
Yeah, go ahead, please.
Okay. Also, one thing for summer study group plans, Madeline isn't able to join. She, like Rob said, is on line 132. She has a non-verbal, or Achintia told me it's non-oral because technically verbal would mean I'm not using words. So, whoops, schooled. But anyway, so it's non-oral updates, and Madeline says, I'm sure he appreciated that I told everyone that too, and we recorded it. Yeah. Anyway, Madeline has a really great summer program because the U of T coders kind of continues throughout the summer. It's mostly run by grad students who have to be there all summer anyway. And so they have a bunch of like summer programs, but if you want to make one of your summer programs just getting together and watching other people's recordings, you could look at their Hangouts on Air. They have a lot of cool recordings. You could all sit around with popcorn and watch somebody else teach a lesson if you wanted. Sometimes that's fun. And for the tutorials and tools and things, these are just things about Julia, not specifically BioJulia. If you want to read up more, there's some interesting blog posts there, and some cool podcasts. I often listen to podcasts for things that I don't use regularly, just because if you hear the same terms repeated like 1,000 times, suddenly it feels like you're, safely like parlant in whatever that the lexicon of that language. So sometimes it's fun to learn that way. And for the announcements, Rob, if you don't mind me giving the announcements, because
I was just about to turn it over to you for those.
Oh, sweet. Okay. So the global sprint that Rob mentioned before, we're super excited. We have over 30 sites. So you should check out the website because there's a sick map and there's these small multiple maps that show you where all the sites are, and every site has its own website. And you can also join virtually too. So if you want to set up your own, but you don't have an official site, you can just sign up to be a virtual participant just so that we know where you are and what time zone you're on, and we can check in with you on IRC or on Gitter and say like, hey, how's your sprint going? What are you guys working on? So please do that. And then another big announcement is that our Mozilla Fellows call for applications opens on June 3rd. So it is like an epic week for the Science Lab. We have the global sprint on the 2nd and the 3rd. We have our website redesign launch on the 3rd, and we also have the CFP going up on the 3rd. So hopefully things won't just crumble in shambles on the 3rd when like the black hole opens of all the stuff we have to do. But I'm sure it'll be okay. And you should apply to be a fellow. Richard, who just spoke about BioJulia, is one of our fellows this year. He's awesome, as you've heard. He is also available for any emails you want to send or tweets about his experience. No, I'm just kidding. I don't want to volunteer him for that.
Yep.
But
No, I am.
but if you want to reach out to Richard about his experience, please do. Our new site URL is going to be science.mozilla.org. So we have like an epic propend domain on the real mozilla.org site, which is rad. And that's going, sorry, I'm very enthused about all these things. And whoever said the open data primer, do you want to chime in about that? Oh, Steph.
Yeah, so I just wanted to mention that Xana and I have been working on developing some really basic primers that explain open data and all the things that surround it. And we're planning currently a series of 7 primers. We're also going to develop some modules and teaching sessions off of that. But the first primer is available for review. It's on GitHub. Please do go take a look. Let us know what you think. We would really love to hear it before we reveal everything to you. Primers 2 and 3 will be coming out within the next couple of weeks. 2 is on how to make your data open, and then 3 is on how to share it. So would love anything that you guys could provide in terms of feedback on that.
Really cool.
Awesome. Yeah, so go check that out. And I always goof up the updates from the last call. These are just here for our benefit, right? Yeah.
Yeah, I did want to spend a second to talk about, so I don't know if there's many study groups on the line. I think some people had to be nonverbal and weren't able to listen, so I don't know how useful this will be. But for the past like 2 study group calls, I've been promising that I'm going to figure out a way to get all of the events from everybody's forked issues from the study group repo and pull it into this epic JSON and then make a rad map out of it. And so I just want to do a little, like, show a little preview of, let me see if I can get the Hangouts on air to stop screen sharing and screen share with a new thing. Let's see. Oh, wow, that's crazy. When I stop screen sharing, it inverses the entire screen and everything's backwards. It's very weird. Okay, so I'm gonna try to find, okay, so here it is. Let me see if I can share it. Yeah, so, okay, so here we are. So this is just a little repo that we're going to be open sourcing to about how to crawl all of the events. So if you're not familiar with the study group project or how events are stored, it's like a Jekyll blog, and in the posts directory, that's where you log in a header, you log the like the basics about the event you're going to host, and then you add it as a post, and you also add it to your issues so that people can have a comment string after after you've posted the event. And then that goes live on your website because it's a Jekyll blog. The events log as blog posts basically, and so you can list events and have a lot of cool details and a whole conversation built around them. So yeah, so the goal of this was to pull those. Everybody forks the repo, the base repo. So there's 180 some forks, which is awesome. And I'm very excited that we have that many study groups, but we don't have a way of letting everybody know all the study group events across forks. So this was just meant to go get those post headers and pull them into a file. And right now I have it working so that it pulls in 100 just as a test case. It doesn't pull in, or there's a lot of to-dos at the bottom of this that say something like, oh yeah, I need to figure out how to deal with when people fork the repo and very sadly never host an event, and I have to control for those errors. But anyway, so that is progressing, and very excited about that. So I'll be open sourcing that later, and I'll make a cool map, I promise. It's not Julia, it's just Node, but I thought that would be okay anyway, because nobody really minds, right? Nobody cares. Okay.
We care.
Oh yay, everybody cares. Okay, I'm gonna go back to the Etherpad now. I think this is, yeah, okay, screen share. So so yeah, that's finally happening. Isn't that awesome? It's very exciting. And recording sessions, okay, so this is the 3rd time we've tried to record sessions, and I'm really hoping that when I close the window, it will all have uploaded perfectly, and everyone who joined at the Hangout on Air, I heard no complaints on Twitter, so I feel like it must be okay. And I also want to say our study group newsletter, we have a Mozilla Science newsletter that we put out every month, or we try to. And I usually like to feature a study group every month. So Rob, I might force you to do this for this month, but I'll give you all the details. Don't worry. And so we have a nice little list of Madeline and Ancon and Nifris, who are the last 2 being champs. Madeline's also a champ because she joins in the middle of her lab meetings sometimes, but the last 2 are on a time zone that is really difficult for this call. They are in Bangladesh and further afield, so they usually have to join at 11 p.m. their time and somehow find great internet, and props to them. So they were featured on the last few newsletters, so that's cool. And next call is June 17th, and Jeff is going to be hosting from Rhody RStats. So so that's rad. So stay tuned if you want to volunteer to MC a future call. Rob did a very good job. Good job, Rob. Oh, that rhymes. Oh, that's so great. I bet people tell you that all the time, though. It's probably really annoying. But anyway, if you want to sign up to MC a future call, please do on line 192. And that's all I have. Rob, do you want to wrap it up with anything?
Yeah, I think we should just thank Ben and Richard for talking about BioJulia. Thank you guys so much. Thanks, Aurelia. And I think that's it. So we'll see you guys next time.
Bye, everybody. Thank you.
Thanks, guys.